So yesterday we finished up, we were talking about this model of purifying selection. And we basically had the parameters, right, n. Our mutation rate was divided into neutral and deleterious mutations. And our distribution of selective effects just involved deleterious mutations with effect SD, right? And we talked right at the end about this intuition that was presented by Brian Charlesworth in the early 90s, basically saying that if you think about individuals within a population evolving in this very simple model, all of them will have been fairly recently descended from a smaller subset of individuals which don't have deleterious mutations, and therefore we can think of the population as sort of effectively smaller. And the sort of more deleterious mutations there are, the sort of smaller it is, and that should be the primary effect of these deleterious mutations on the diversity within the population. Basically the idea being less diversity if there are more deleterious mutations, because that corresponds to a smaller effective population size. Yeah?
I don't quite follow that, because I would imagine if there are more deleterious mutations that would affect your fitness function. But why does that affect your effective population?
Well, okay, so let's just talk about it now in a little bit of quantitative detail, okay? Because I'm going to sort of, why don't you ask me that question again if it's not clear in 5 or 10 minutes, okay? So let's start in analyzing this model by thinking about the distribution of fitness within the population. Okay, within the context of this very simple model, okay, the individuals can be categorized according to how many deleterious mutations they have, because all the deleterious mutations are equivalent, right? And so I'm going to define HK as the fraction of the population that has K deleterious mutations. And if we think about the sort of continuous time approximation for a Wright-Fisher model, what we'll see is that the fraction of individuals that have K deleterious mutations is going to evolve according to this. Okay, according to this equation. Okay, here, just for the sake of making the notation a little cleaner, what I'm going to call these deleterious mutations, their effect is some minus S. Okay, so S being a positive number, just for the sake of keeping the signs that look a little bit clearer that way. S is some positive number. It's how costly is a deleterious mutation. So the fraction of the population that is K deleterious mutations, it's if you have this K bar is the average number of deleterious mutations for a random individual. So if you have less deleterious mutations than average, K is less than K bar. This whole thing is positive by how much more fit than average you are. Or if you have more deleterious mutations than average, then this whole term is negative, right? You're either growing or declining exponentially according to how many deleterious mutations you have relative to the mean. This is just saying, well, you are acquiring deleterious mutations at rate UD, right? And so the fraction of the population that has K deleterious mutations is declining because some of those individuals are acquiring additional mutations. They get to K plus 1. At the same time, it's increasing. There's individuals that have K minus 1 deleterious mutations, and they're acquiring new ones so that you have K. And then there's genetic drift, which leads to a noise term. Okay. And for the case of this simple model, okay, in many of these models, and we'll talk about in a few minutes one that doesn't have such a nice property. But in this particular model, there does exist a steady state solution in a sort of n goes to infinity limit where we can neglect noise. Okay. So let's talk about that briefly. If we neglect genetic drift, okay, we think about sort of a very large population where genetic drift is negligible compared to these other terms. This is just a system of differential equations, okay? It's nonlinear because of this K bar term, okay? But we can solve this, in fact, exactly. For arbitrary initial conditions, one can get the full time-dependent solution. Okay. I'm going to leave that as an exercise because it's not really important for what we're going to talk about today. But we can also just very easily write down what you get for the steady state solution. Okay, so in steady state, we'll get some stable distribution of hk by setting this equals to 0. And what you find is that in steady state, we have this becomes some Poisson distribution. Okay, all right, where lambda I've just defined is ud over s. Okay, so what this is saying is that the average individual has lambda deleterious mutations, and the distribution of the number of deleterious mutations is Poisson with that mean. Okay, and one particularly important thing is that the fraction of individuals that have no deleterious mutations, just plug in k equals to 0 there, and I get e to the minus lambda. So what does this look like? If we just plot this, okay, obviously it depends on what lambda is, but this is going to be some Poisson distribution, whatever. This is k equals 0. I draw this like this way around because this is high fitness, this is low fitness, okay? So it's kind of backwards a little bit, but whatever. You get the idea, okay? Right? So the idea that Charlesworth had, essentially, okay, is that if we sample individuals from within this population, right, take two individuals, okay, from within the population, they're sort of rapidly descended from individuals in this 0 class. All these individuals in the 0 class are selectively equivalent. So the evolutionary dynamics within this class are essentially neutral, right? And so we can think of this as a sort of neutral population, okay, with size n h naught, which is n e to the minus lambda, okay? And he said, right, let's call that a sort of effective population size, okay? And so these two lineages, this happens very rapidly, he argued, okay? And then when we look within this class, okay, the dynamics are essentially neutral, and so there's just neutral coalescent theory, okay, within this reduced population, okay? That's the argument that was made, and is, you know, if you read textbooks on population genetics, what they'll say is that, you know, this is the answer. This is what purifying selection does to diversity. Does this answer your question from earlier?
Yeah.
Yeah.
I'm still the effective population size should not just be a function of the one with 0.
Right.
But I roughly get the idea.
Yeah. So this is the intuition, but you're absolutely right. Okay. So just to give you an example of a problem that you know my lab has worked on, a few other labs have worked on, we've sort of had the same dissatisfaction that you were just articulating. And there's various things that are like obviously screwed up about this. Okay. So first of all, I mean, can we just neglect this compared to, you know, these times compared to this? Okay. And the answer is sort of, yeah, you can, provided that NE is approximately equal to N. Okay. So in other words, this is a good approximation when it doesn't matter. Okay, is a sort of way of putting it, right? Whenever purifying selection does significantly affect diversity, okay, then this can't be a particularly good approximation. We can sort of see that just based on a very simple argument. Like, let's say, you know, we're trying to compute the site frequency spectrum, right, that we talked about before, okay? The number, and not just the site frequency spectrum, but actually the total number of polymorphisms that are at frequency x, okay, within the population. Remember that yesterday we talked about how in a neutral model f of x is basically 2nu over x, okay? So what this predicts is that there's some sort of NE which is reduced. Okay, so there's some NE which is reduced, and this goes like 1 over x. Okay, I'm trying to draw that. It's probably pretty bad. Okay, and So the idea is that there's some, reduced NE, so this whole, this would be without purifying selection, I should say, right? And then with purifying selection, everything shifts downward. We still have the 1 over x behavior, but the sort of effective n is lower, so just everything is shifted downward, right? And if you think about it, like there has to be something really wrong with this, because as we talked about, maybe 2 times ago, right? If n is, for mutations that are really rare, right, such that their population size is small compared to 1 over the selective effect, right, their dynamics are essentially neutral. They can't feel the effect of selection until they get big enough, right? And so what that means is if we think about sufficiently rare mutations, they can't possibly know about the effect of natural selection. And so you have to go to this at very low x. Okay? And so, maybe this is true for common mutations, but then there has to be some kind of transition, right? In which case, like, this can't really be right. All right? And basically, this is a signature of the fact 2 things. Well, actually 3 things. Okay. Thing number 1. This time is not necessarily negligible. Okay? In fact, typically it won't be negligible. Okay? And that's going to distort the structure of the genealogies, okay? And hence the diversity within the population, because these things are now no longer obeying the Kingman coalescent, okay? Because they're not selectively equivalent. Thing number 2. You can have coalescence among individuals with deleterious mutations, right? Like I could get some individual from this class, gets a couple of deleterious mutations, and then leaves lots of offspring just by chance. And if I sample those offspring, well, they'll coalesce somewhere here. Okay. Thing number 3, this whole deterministic approximation, okay, if I look at it, okay, it's a deterministic approximation. And so, there's just some obvious things that are sort of pathological about it. Okay? Take, for example, okay, this size, okay, this is the size of this 0 class. Okay? It's the population size times e to the minus lambda. Okay? So this is exponential in lambda. Okay? So even for very large n, it's not too hard to imagine this getting really small. Okay? And if this gets really small, then genetic drift will matter, okay, even in very large population sizes. Okay? And what can happen in this model, right, the genetic drift will cause this to fluctuate. And then as this fluctuates, if this gets bigger, that'll sort of lead to sort of propagating fluctuations through the rest of the distribution. Right? Because if this gets bigger, well then this term sort of gets bigger for a lot, for the H1 class and tends to make H1 get bigger in some time delayed fashion. There's a whole complicated process of propagation of fluctuations through this distribution. Okay? And most dramatically what can happen is this thing can go extinct. Okay? And once this class goes extinct, this model is somewhat artificial in this sense, but within the context of this model, it can never be replaced. Right? And so the whole distribution has to move left. Does anyone know what this process is called? Yeah. Yeah. So a lot has been written about this. It's called Muller's ratchet. Okay? And so, here I'm talking about diversity. Okay? Within the context of this model, of this approximation, all right, by definition, this class doesn't go extinct. They're always individuals with no deleterious mutations. That implies that P fix of S is 0. All right, and that there's no accumulation of deleterious mutations. But clearly that isn't the case. Right? And you can see like weird things happen. Like say I send S to 0. Like as S gets close to 0, okay, this gets exponentially small. All right, this predicts the diversity is getting less and less. But obviously as S goes to 0, those deleterious mutations are no longer deleterious. And so somehow we should go back to the original neutral prediction, right? Which we're clearly not doing here. Okay? So this remains a topic of active work. So still, like, nobody can really predict what the diversity should actually look like in the presence of all of these complications. In particular, in the presence of this noise, okay, we just don't know, okay? I mean, there's folks in my lab who have, published a few published papers on this. There's some things we know, but in general, we just don't know, okay? Couple of things we can say within the context of this model, so long as lambda doesn't become too big, so we can still make the approximation that we ignore noise here, all right? We can account, and I don't want to take too long on this because I want to get to experiments, but we can account for these other effects of coalescence occurring somewhere in here, as well as the time it takes here. And what we find is that to at least a reasonable approximation, we can approximate the evolution as being the same as in a neutral population, but with a time-dependent population size that looks kind of like this. Okay? And I mean, the intuition here basically is that it's the same kind of idea that Charlesworth had, right? Which is that as we look backwards in time, the individuals in the present are descended from a smaller and smaller and fitter subset of the ancestral population. But now no longer saying you're sort of quickly descended just from these guys, but then, as you go backwards in time, one can think about one can sort of quantify, as you go backwards in time, what is the size of the smaller fraction of the population that you will on average be descended from, okay? And based on that, one can compute this effective population size, okay? And you can see that, like, as for t equals to 0, this is just the whole population. And as t goes to infinity, this just becomes n e to the minus lambda, okay? But there's a whole transition in between, okay? But this result, I mean, this is not the greatest approximation in the world, but it highlights a sort of important point that I've been sort of talking about before, which is this sort of problem that it's sort of too easy to fit things, fit observables in different models. There's a problem of identifiability, right? So yesterday we talked about fitting demographic models with time-dependent population sizes to, say, human data, okay? And finding population size histories where, the population was smaller in the past and is larger in the present. And we showed a figure of the fish, right, which show exactly the same thing. And any kind of population that you look at tends to show this same pattern, okay? And in some cases, we actually believe the population size was small in the past, okay? But what this is showing is that just purifying selection by itself in the context of this absurdly simple model will lead to an apparently time-dependent population size, at least from the point of view of predicting what the site frequency spectrum looks like. So finding, fitting those demographic models and finding a population size that was smaller in the past, it could mean that the population was smaller in the past, or it could just be a signature of this process, right? And so, to me, that's sort of an interesting, an important observation for the interpretation of data. Did you have a question?
Yeah, some terminological intuition about the 1 minus e to the minus t whole squared.
Yeah, so basically what's going on here, the way that we calculate this, okay, is that we think about the ancestral fitness distribution, okay? So if I pick one individual, okay, it comes from class K with probability HK, okay? And so now a natural thing to do is ask, what is the distribution of the relative fitness of its ancestors a time t before the present, okay? And you can basically compute, because if you think about backwards in time, let's say I'm in class K, okay? So what class was my parent in? Okay, so class K has size NHK, okay? And every generation, a fraction, a number, sorry, NHK minus 1 times UD individuals came into class K from class K minus 1 due to the acquisition of a new deleterious mutation. So every generation in class K, this is the fraction of individuals that just came into class K via a new deleterious mutation. So backwards in time, if I think about it, if I'm in class K, this is the probability that one generation ago I was in class K minus 1. My parent was in class K minus 1. And with probability 1 minus this, my parent was in class K. Right? And so based on that, you can calculate basically the probability that at the time of sampling, the present, you were in class Ki, okay? And then that backwards in time, your ancestor was in class Kf a time t generations in the past, okay? This is just a sort of, Poisson process backwards in time, okay? And we know these HKs, okay, assuming this noise-free is a good approximation, and we get this equals e to the minus skit, e to the st minus 1 to the ki minus kf times ki choose kf. Okay, that turns out to be what you get if you do this calculation. So then what we imagine doing is you just pick two individuals at random. The ki's are drawn from hk because it's the present, right? And then you think about backwards in time, what is the probability that they're in these classes, okay? And that gives you a sort of the subset of the population that each is descended from, right? And that sort of tells you what the coalescence probability was at that time. And that's where this is coming from. Okay, where this is coming from, okay?
You didn't account for the probability that you were in class K and then you went to class K plus 1.
Yeah, but backwards in time, it's the other way around.
Yeah, but then the probability that you were from class K, given that probably that your parents were in class K, given that you are currently in class K, will not be 1 minus the probability that your parents were in class K. Okay, I'm very confused. But yeah.
No, right? There are some number of individuals in class K. You're in class K. Every generation, some fraction of the individuals in class K minus 1 are newly in class K minus 1, right? And so the probability you are newly in class K minus 1 is just that fraction. If I just sample you today, and so if you're in that fraction, that means by definition your parents were in class K minus 1, or your parent was in class K minus 1, right? And if not, then not, right? And so that's all that is within this model. Okay. Okay. I mean, what I'm trying not to do is we have a paper on this which you can read and it goes through all the calculations and whatever. I could spend a whole lecture on this, but I don't want to. But I just want to give you a sense of the kind of things, the kind of calculations that you can do now, and the sorts of questions that are open. So one of the things, now I really am going to make you dissatisfied a bit, because one of the things that we've been working on lately is, and we just published a paper on this just last year, another way to think about this problem is just forward in time. Okay? So you remember before, right, we talked about, the probability, from these diffusion approximations, the probability that a given mutation has frequency x at a given time. And if we could compute what this distribution looked like, then we know the site frequency spectrum, because basically the fraction of polymorphisms that are frequency x is the site frequency spectrum, right? And so the question is, could we calculate this forward in time in this model? And the answer is, it's really difficult to do, okay? Because basically, if we think about, let's say a new mutation arises, okay, and it has k deleterious mutations. So we can model its dynamics using either a branching process model like we talked about maybe two lectures ago. But as it accumulates additional deleterious mutations, you have that stochastic branching process, okay, of the size, the number of individuals, in class K that has this mutation, but then that starts to feed mutations into lower classes as additional mutations accumulate. And one needs to compute the sort of joint frequency of all of those things, because you have, I have some number of individuals. Okay, let's say I get a new mutation. Let's say I get a new neutral mutation, okay, because there are neutral mutations in an individual with K deleterious mutations. Okay. And what is its P of X and T, or I'm going to talk about the P of the number of individuals in T, right? So, it has some dynamics, okay? Goes up and down according to some branching process model, which we can write down. But these individuals can acquire additional deleterious mutations, right? And so we sort of have to think about, let's say a mutation occurs, a neutral mutation occurs in one individual here that has 2 deleterious mutations. That starts to generate a lineage that has 3 deleterious mutations, and then mutations occur here. And these things are all fluctuating in some complicated coupled way, okay? And so, if you're interested in how to sort of analyze these dynamics, all right, we were, able to do so essentially using a sort of approximation where we treat this sort of initial class stochastically and then treat all the sort of things that are happening as new deleterious mutations arise as sort of slave deterministic behavior that's just, slave to the stochastic dynamics here, okay? And I don't have time to really go into all of the details, but just to give you a sense of what you wind up getting, okay, you get the following site frequency spectrum. This allows you to calculate the site frequency spectrum sort of analytically. Just to show you the kind of stuff that you get, here's what we find. Okay. This should all be completely intuitive and obvious to you, by the way, if you're paying attention. I know I saw these things for the first time and I was like, yeah, that's clearly right. Okay. All right. So this is what you get. All right. So I don't have a pointer here. Here, sigma is just the standard deviation of this fitness distribution, right? It's a Poisson distribution. It's standard deviation. It's obvious from there. Okay. So what we see is that for very small x, we have that neutral site frequency spectrum, 1 over x, same as, for the neutral population. Okay. As x gets larger, we get something that's approximately 1 over x squared, a steeper site frequency spectrum, okay, with some log correction. Okay. Then for even larger x, okay, we go back to a neutral site frequency spectrum like 1 over x, but you notice that sort of the n has been replaced by n e to the minus lambda, right? That's the sort of effective population size that Charlesworth was talking about. And then a kind of funny thing happens for even larger x, okay? Actually the site frequency spectrum starts to go back up, okay? And then, well, you can't see it, this is again 2nu over x. For very large x, we go back to the original neutral site frequency spectrum, okay? And so basically what that looks like, drawing it here, okay? So x can go between 0 and 1, all right? Initially, we get this 1 over x behavior. Then we get a sharper 1 over x squared behavior. Then we get a 1 over x behavior. And then we actually go up again, and we asymptote to sort of what the 1 over x behavior would have been all the way across. Okay? And it's kind of funny to me that, like, I mean, this is not really rigorous in any way, but let's go back over here. I don't know. To me, it looks kind of suspicious. All right? I mean, it is sort of what these site frequency spectra look like. I mean, now obviously there's a lot of stuff wrong with this, right? I mean, this is I don't obviously believe that reality is this simple model, but it's kind of intriguing that you can get like all of this behavior from the simple model, and it's not at all intuitive. Like, just to give you an example, right? This uptick here, I mean, people think it could arise, you know, in this kind of data from just artifacts of ancestral site misidentification like I talked about before. But the other thing that people thought about, well, this just has to be positive selection to the extent that it's real, right? These are mutations that are beneficial. And they arise and they're sweeping through the population, and that's why you get sort of more of them, because some small fraction of mutations is beneficial, and you tend to see them at high frequency. And you know, that may be true. I mean, certainly beneficial mutations would do that, but it's sort of interesting and counterintuitive to many people that you get the exact same thing out of a model of purifying selection. And this is like actually reflective of a sort of real deep symmetry in population genetics between beneficial and deleterious mutations, right? There are differences, but they're really subtle. You remember when we were talking about the branching process models, right? Where the dynamics is basically neutral below 1 over s, and then, you know, for 1 over s, if you're beneficial, you sort of go up, and if you're deleterious, you bounce off, right? So the difference between going up and bouncing off is obviously a difference, but like the dynamics are still like really primarily dependent on the absolute value of s, okay? Not on whether it's negative or positive. And you know, here we're getting this sort of uptick, because if you think about it in this model, right, within the population at any given time, some individuals have more deleterious mutations than others. And so, like, you know, there are these guys that have one deleterious mutation, right? And if you think about the mutations that they have, right, these guys are more fit than average. So whatever mutations they do happen to have, you know, they will look like they're positively selected. Because they are, right? They're positively selected not because they're good, but because they happen to be in genomes that are less bad than average, okay? And so, like, actually, if you think about like the signatures that one would expect to see in genetic variation, any kind of signature, it's going to look like those are beneficial mutations, okay? Even though they're not. All right, so that's another sort of important caveat of the sort of problems with identifiability. Now, obviously, there is a lot left to do here, right? We might want to think about, well, what happens as there's a real distribution of selective effects? This whole approximation, you know, obviously, just like this earlier thing, like if n e to the minus lambda gets really small, such that this distribution is not stable, there are fluctuations, maybe there's Muller's ratchet, okay? That we don't understand. Obviously, somehow as s goes to 0, the dynamics have to go back to neutral, right? Whereas this sort of predicts bigger and bigger distortions for smaller s. So there's some other side of that that we don't begin to understand. This is also not including a realistic model of recombination, right? Right now we're just thinking this is a linked, and this is another sort of thing that I think is a big open question, right? Last time we talked about how, we're thinking about evolutionary dynamics within some linked region of the genome, right? Where the sort of length of that linkage block depends on the time scale, right? The linked regions are the regions that stay linked. There's not likely to be recombination on the time scale at which common ancestry is determined, right? These coalescent time scales. But if you think about it here, the coalescent time scales, they depend on the selection, right? They depend on, say, UD. The bigger the UD, the shorter the time scale, right? And so, but then the shorter the time scale, the bigger the relevant block, which implies a bigger relevant UD in a linked region, right? So there's some self-consistent thing that needs to be thought about there that's not really well understood. Lots of questions like this. All right. So there's a lot of open things to do here. And before I get into experiments, I just want to then very briefly here say a couple of words about the opposite model. Okay? The opposite model, which is actually a model I started working on well before thinking about this. Okay? Instead of having deleterious mutations, we have beneficial mutations. Okay? So instead of having some distribution, we just imagine one is a positive S. Okay? And you can imagine trying to do the same thing. Okay, now this just becomes a plus. Okay? This becomes a UB. Otherwise, everything is the same. Right? But in contrast to the model I just talked about, now there is no solution to this equation in the absence of noise. Okay? If you think about it, it's sort of obvious, right? The distribution is, you're going to accumulate beneficial mutations, so there's not going to be a steady state, right? You're going to go to higher and higher K. Nothing's stopping you. And so, a sort of thing that we started thinking about, this was like, 15 years ago when I started getting into this, is, okay, there's no steady state distribution that we can write down, but we might imagine that there's some kind of traveling wave. Okay? Because what's going to happen is, new mutations are arising, driving you this way. Selection is killing the less fit than average individuals. So you might imagine there's some traveling wave solution. Okay? And in fact, there is. One can try to write down, look for traveling wave solutions here. But it turns out that you can't even find a traveling wave solution in the absence of noise. And the basic reason is that if you think about it, as you sort of push out this way, these classes are growing exponentially at sort of faster and faster rates. And so the only traveling wave, deterministic traveling wave solution you'll get, okay, has v going to infinity, the rate at which you move going to infinity, which obviously doesn't happen. Okay? And the reason is basically because of finiteness in population number, even in arbitrarily large populations, there are a few most fit individuals within the population. Okay? And it's really the dynamics of those most fit individuals that drive what happens. Okay? And so, I want to get into the experiments, so I'm not going to sort of talk about the calculations. But we were able to calculate things like P fix of S, the corresponding V, okay, and the site frequency spectra in this model, okay, by properly accounting for these stochastic dynamics. And just to give you an example, what we find is stuff like this, all right? Which basically implies, okay, that, and here, let me write down the corresponding P fix if it's useful. So that corresponds to a P fix is just two log of ns over nu log squared of S over U. Sorry, all these are natural logs. Okay. Which basically implies that fixation probabilities and the rate at which the population changes in fitness, instead of depending linearly on U and N, like we talked about before for the model where every site is independent, they depend only logarithmically. Okay? So this is sort of a much slower adaptation. All right? And I'm not going to get into how to calculate this, but anyone who's interested, I can refer you to stuff to look at. And one can correspondingly then, do these kinds of things of tracing lineages through the distribution to compute things like the site frequency spectrum. You had a question in the back, yeah?
I think I lost why you started to consider this model just neutral.
So basically, ideally we want to consider models that include everything, right? The sort of intuition is that, I started doing this like 15 years ago, and I was looking at experiments, which I'll tell you about just starting in a minute. And, in these experimental populations, they're clearly, the dynamics is driven by beneficial mutations. They're adapting. And the primary thing that's happening is they're increasing in fitness due to accumulation of deleterious mutations. So it was natural to consider models that focused on beneficial mutations. And then if you imagine a population that's sort of well adapted to its surroundings, you might imagine that there aren't really very many beneficial mutations, and you primarily just have purifying selection. And so natural to consider that alternative model. Now, obviously, reality includes both, right? And I was talking about in the first lecture, right? Like, you might expect actually like a real situation to include, V is basically 0. You're not going anywhere, but there's some beneficial and some deleterious mutations, and somehow they're in balance, right? And those are things that are just not understood, right? But like, it would be a natural thing to do, and a sort of thing that we're trying to work on, and other people are trying to work on, a model that includes both beneficial and deleterious mutations with a range of effects. And that's really what we should be driving towards. The hope is essentially, and this remains to be seen whether this is true, but the hope is that by considering these very simple models, we can do two things. One is just demonstrate that certain kinds of things that people think are interesting are not, right? Like stuff like this, these distortions, to try to, lay out the landscape of identifiability, right? And for that, all you really need to do is, like, if you can show that in this simple model you can get the same result as some time-dependent population size, that's sufficient to show that the data doesn't distinguish, right? And so that's important. But then the third thing is the hope that, by developing the theoretical methods to start analyzing these simpler models, we can then start to put them together and, build on them to get into more complicated scenarios. And whether or not that's going to work, I mean, it'll be very case dependent. It's not clear, but that's a direction, right? Yeah, and from there you can start adding on all the kinds of things that you guys were bitching at me in the first lecture for not including, right? But hopefully by now you sort of see that, like, even though I've neglected a lot, there's still quite a lot to do. Yeah? This is the probability some random new mutation will eventually fix rather than going extinct, right?
So if you have like no steady state or no traveling wave,
there is a traveling wave. It's just to calculate what it looks like. It's sort of, you cannot neglect noise. Because the population isn't actually running away because there's finite size effects, right? And so even an arbitrarily large, right, like often in these traveling wave models in physics, you'll compute some steady state, n goes to infinity behavior, and then treat, 1 over n effects as a perturbation. And here you just can't do that. But there is still a traveling wave. And there are still, each new beneficial mutation, some of them will fix and most of them won't, and right? And you can sort of see here, right, in the absence of this, if every site was independent, we saw the fixation probability was just S for NS large. And now it has these log factors, and maybe it's not obvious, but these log factors are small, right? Because you have these log factors divided by 1 over NU, okay, for large N, this will be a small factor. So that, basically this reduces the fixation probability of beneficial mutations and slows things down. Okay, it's a constraint on adaptation. All right, and hopefully the reason for this will become a little also clear in the experiments. All right, I have 45 minutes left, so I want to at least give you a flavor of the kind of things we can do experimentally. In a sense, this experiment stuff is even more. I could talk for even longer about this, but I'll try to sort of give you a sort of overview of what kinds of things you can do and look at. All right. So as I mentioned, we do these experiments. Do I have something that works? Oh yeah, sort of. All right. We do these experiments I primarily work in yeast in my lab. Other people work in bacteria. I'll talk about experiments in both. Okay, but basically, there's a variety of ways you can maintain the populations, but basically they boil down to, you put some bugs in some sterile media, you let them grow, and then you dilute some small fraction into some new sterile media, and you just repeat this again and again and again. Okay, you can tune the population size by basically tuning, how you do this dilution and how big a tube you use, And you can also tune things like, you can change the mutation rate, okay, by either putting mutagenic chemicals in the media or by knocking out mismatch repair genes in your bugs. People do these experiments in Drosophila where you can't freeze them, but in bacteria and yeast, we can just freeze these things indefinitely at -80 and in 20% glycerol, and so then you can go back and look at things. The fitness here is something that's actually quite straightforward to measure, although we'll see some caveats to that in a minute. It's basically if you have some genotype that is present in this population, its fitness relative to the mean of the population at the time is on average, if you start with one of those individuals, how many will it leave in the next dilution cycle, right? And that includes various things about lag phase growth and saturation phase and exponential phase. It doesn't really matter. The point is that if you leave on average more than one offspring, then you're more fit. If you leave on average less than one offspring, you're less fit than average. And we can measure this, and in practice what we do, we use these robots so we can do a lot of stuff. Simultaneously, we can do thousands of populations at once using these robots to move the liquids around. And to measure fitness, like say you have some evolved genotype, evolved clone you want to measure the fitness of, we have these fluorescently labeled reference strains. We just mix them. You can measure the ratio, and you can just then propagate them for a couple days and measure the ratio again, right? And if on average this guy's leaving more than one, right, the difference in these ratios will tell you about that, right? Obviously, there's some noise associated with these measurements, but that's what you sort of get, all right? So, we can do these kinds of experiments, all right? And, this is a typical thing that we see in our yeast over the course of 1,000 generations of evolution. You start by definition at the ancestral fitness, and then if you look at the evolved strains over time, they increase, In this particular case, this is a situation, we did 600 replicate populations. These are the average fitness of those 600 evolved populations after, however many generations of evolution compared to the ancestor. These error bars, they're not the errors in the fitness measurements, but rather the sort of standard deviation between, among the 600 populations, right? By chance, some of them evolve faster, some of them evolve slower, and so you see that. And so there's a variety of ways that we can start to look at the dynamics within these populations. So here's a thing that until recently this has sort of been the best way that we've found to look at what's going on in these populations. Basically the idea is, you take frozen samples from the whole population at a variety of time points, okay? And you just grind up the whole population and you sequence it. Okay, and any mutation that's arisen, let's say there's a mutation that's present in 30% of the individuals at that time point, then you'll see 30% of your sequencing reads at that site support the mutation and 70% don't. Okay, there's obviously a resolution limit to this associated with the details of sequencing error rates, as well as all kinds of actually dominant effects can often be bioinformatic alignment artifacts and things like that. But the bottom line is we have some frequency resolution, which I'll talk about right at the end, ways that we're trying to do a lot better than this now. But like, this is an example of the kind of thing that you see. This is one population. Okay, these are all the mutations that we observed that reach a frequency of at least, say, about 10% at some time point. All right, and this is their frequency over time. And you can see all these sort of complicated dynamics of interference and so on. This is a sort of alternative way. I haven't showed these yet, have I? This is called a Muller plot. So the idea here behind this Muller plot, I'll show more of them, is that we're sort of imagining lining up the individuals here, and this is time. And so it's just an interpretation of, you can infer this from this, basically. Some individual got a mutation in this gene, some other individual got a mutation in this gene. Somehow some lineage got, this combination of 6 mutations. We don't really have the resolution to see exactly what happened. Presumably one at a time happened. But and this lineage got 3, right? These mutations happened within an individual that had this one, and so on, right? And it sort of drives this. And you're going to see, if you think about it, right, these mutations, these populations I haven't mentioned, these populations in the lab, they have millions of individuals. So by the time you get to 10% frequency that we're observing them, you're talking about hundreds of thousands of individuals. Okay? These mutations have selective effects on the percent level, okay, plus or minus. And so what you're seeing is essentially all of these mutations have been very successful. They've survived that genetic drift while they're rare. They've gone well above that 1/S threshold. And, for instance, this mutation would definitely fix if it weren't for these things. Okay? But this process that you're seeing is a sort of competition between all these competing mutations in different lineages. Maybe not all of them are beneficial, but certainly a substantial number of them are beneficial mutations, and that sort of playing out, right? These guys getting lost is essentially, this factor of mutations that have sort of survived genetic drift and grown up, but now are getting competition from other mutations. All right? And we see, these are half a dozen examples, 8 examples. And what we would like to do essentially is understand these dynamics, with this sort of, here's a sort of schematic of this traveling wave model, right? Now, in reality, of course, we don't have just a single selective effect, but we have some traveling wave distribution of fitness within the population. New mutations are arising, they're spreading, other populations, other mutations are happening. Here's an example of a mutation that happened and sort of got left behind. Okay, and so all these dynamics that we're observing are a sort of signature of these processes here. Okay, and so yeah. These don't mate. So, although the reason, the things I was just showing you are purely asexual, right? So it's like these yeast have about a 12 megabase genome. So like we're looking at, the dynamics of a linked region of 12 megabases, essentially. Now, one of the reasons I started working in yeast is because you can sort of make them have sex on demand, okay? It turns out actually to be quite complicated and painful to do so, but you can. And so like we have other sorts of experiments where we're having them mate at different frequencies and look at what happens. When we do. I'm not going to talk about those, but just because there's only so much, I don't have that much time. But yeah, that's a super interesting thing to look at.
So the mutations get shuffled.
Then the mutations get shuffled around. Exactly, right? Yeah. Yeah.
I was wondering, do you sometimes see different combinations of mutations?
Yeah. You see all kinds of different combinations and different replicate populations. We see we see recurrent themes. There are certain mutations that appear to be sort of generally beneficial in the conditions that we typically use. We see them again and again across replicate populations. Some of them, sometimes they succeed, sometimes they don't, just depending on what other, what combinations they happen to occur in that particular population, right? And I mean, there's a huge amount that I could tell you about the details of analyzing these things. I'm not going to do that. I'm happy to talk with anyone who's interested afterwards or point you to things to read. But what I want to do now is just, try in, 40 minutes to give you a sense of the kind of data that we have and the sort of questions that you can ask and what it is that sort of wants to be explained. Okay, yeah.
How interpretable are the mutations?
Well, I mean, they are. I mean, it depends on what you mean by by interpretable, right? I mean, a lot of the things we do have sort of mechanistic stories about what's going on, okay? Other things we don't. And there are certain properties of the things that we don't really, about interactions and stuff that we don't understand. But like, I mean, one of the things that happens a lot in these yeast, for instance, is they turn off, I mean, the way I like to put it is that they learn to trust us. The yeast in the wild is like very risk averse, right? So I mean, or maybe not in the wild. I mean, traditional lab yeast anyway is risk averse, right? Because people have been mistreating stuff, and basically, what they do is if conditions start to get a little bad, they sort of go shields up, they hunker down, they try to do all kinds of stress response, because the sort of thing is like things are getting bad, like they're likely to get worse, right? And essentially, like at some level, what we're teaching them is that, if things are getting bad, don't sweat it. The graduate student is going to come again. It's going to be fine. And so, they turn off stress response. But then there's all sorts of other things that happen. And I guess, though, the sort of, a lot of sort of people with a biological background, that's like their primary interest. Like, what are these things doing? And I guess what my perspective is and what our sort of approach is that we sort of don't want to go too deep in that direction. And the whole point is to like understand what the dynamics look like, what can we say about evolution as a process, and ultimately like what are the sort of frameworks that we can use to try to infer what the selection pressures are and so on. So that, because we don't actually care about yeast in the lab in these conditions, right? Like what we're trying to use this as is a sort of model system to help us interpret observations of evolution in more relevant circumstances. But yeah, I mean, you can't avoid sort of thinking about it a little bit. All right, I want to just briefly talk about, we've spent a bunch of time actually also looking at this really famous evolution experiment that Rich Lenski has done in E. coli. How many of you have heard of this? Okay, most of you. So it's the LTEE. So Rich has been evolving these 12 replicate populations of E. coli, effective population size of about 30 million, 6 generations a day. He's done this for more than 30 years now. At the time we picked it up a few years ago, that corresponded to 60,000 generations. And he had frozen down samples every 500 generations. Okay, this is a long time. Okay, and what you see is this sort of, this is I think like a really kind of nice system. It's obviously one that I haven't been able to replicate yet in my lab. I'm working on it, but the 2035 Boulder School or something, we can talk about it. But 2045 maybe. Anyway, this is a sort of unique resource. In a way, it's like, it is the sort of, I think, unique example of a set of populations evolving in constant environments, is as constant an environment as you're ever going to see for as long as you're ever going to see, right, in some controlled way. Okay. Now, I mean, is the population really exactly constant? Maybe not. But I mean, this is about as good as you're ever going to see. Okay, and these guys, they've measured fitness changes in the way that I described before, and what you see over the first few thousand generations, there's these rapid increases in fitness. They are continuing to increase, which is kind of astonishing, all right, but relative to the ancestor, but it's sort of really kind of slow, okay, beyond this point. And so, in a sense, like, if you're ever going to sort of have a simple model of a population evolving in a constant environment, maybe well adapted to that environment after a while, like this is about it. And so Ben, who is a grad student in my lab, he's about to start a lab at Stanford. You should go work with him, really, seriously. And Mike McDonald, who was a postdoc at the lab at the time, they we basically wrote Rich and we're like, why haven't you done this yet? And he's like, well, do you want to do it? And so I said, sure. And he just shipped us all these samples, right? 60,000 generations, frozen samples every 500 generations. That's 1,440 samples over a dozen populations. And we did this metagenomic sequencing that I was describing. Okay, and here's like what you start to see. So here, this is the first 10,000 generations of one of the populations. Again, right? You start to see just new mutations arise. You follow their frequency as a function of time, blah, blah, blah. Some of them fix, some of them go extinct. New mutations arise. You can follow it along, okay? So we did all 12 of these through 60,000 generations. Here's 6 of them, all right? We screwed up here. Otherwise, it was okay. 60,000 generations. Each of these panels is one of the 12 populations. I'm just showing you 6 here. And each of these lines is a new mutation that arose. Okay, we know what they are. I'm obviously not showing you. We know what genes they're in, etc. But I'm not showing you, just showing their frequency over time. Right? So you can see, right, there's this sort of whole god-awful mess of stuff going on. And there's quite a lot to think about in these populations. So, and this is the sort of thing that sort of inspires some of this modeling. So, a lot of things to think about. So first of all, like, evolution at this molecular level, it really doesn't slow down, right? Remember from the fitness, they increase in fitness a lot over this little bit, and then they kind of level out. You can't really see that from this data. They just look like lots of stuff is going on the whole time. These populations, actually, they all increase in fitness in a fairly reproducible way, but you look like, the 6 populations, they kind of look different from one another. I don't know, like, I don't know, like, what my null expectation going in should have been, right? Are these like remarkably similar or remarkably different, right? But anyway, to me, they look a little different. Here's the other 6. Okay, these ones are really kind of neat because in each of these 6 populations, at some point or another, some mutation occurs. Okay, it's most obvious in that top one. Some mutation occurs that knocks out a mismatch repair gene, and then the mutation rate goes up. And then, from then on, it's kind of a wall of color because there are just thousands of mutations happening. In this case, I believe it's mut T here that fixes. And it's also kind of remarkable to me, like as a sort of, thinking about it, that there are lots of mismatch repair genes. And if you just do back of the envelope calculations, each of these populations gets hundreds of thousands of mutations in mismatch repair genes over the course of 60,000 generations. And they fix in 6 and not the other 6. Right? So each population is getting like 100,000 opportunities to do that, to elevate the mutation rate like this. And it turns out, right, I would have thought like either all of them would become like this or none. Half is kind of weird. And in addition, these populations, right, if you look at the number of mutations they accumulate, they shoot up, but the sort of fitness changes aren't really affected. Yeah, Ilya.
So, another question that I always have about that
Yeah.
slide is why these mismatch repairs haven't fixed in the 50 years that the strains were in the lab before, or even in the thousands, millions years even before that, right? If they got to be fixated so quickly.
Yeah. It's a good question, right? And you know, if you look at E. coli in the soil, right, you will find a kind of either a shockingly high or a shockingly low, right? Like if you look at E. coli in soil, you'll often find like 1% or 2% of them have these hypermutator phenotypes. Like, why is it that amount? I don't know. I just don't know.
Is there some
There's something else that happens in these. Yeah, there's theoretical questions to ask. And you know, so in addition, like some of these, okay, I've sort of inset, this is like the number of mutations accumulated as a function of time, sort of like the number of fixed mutations as a function of time. And here in this inset, I'm showing the 6 mutator populations, right, because you can't show them on the same scale. Some of them, they level out after a while. They actually fix mutations that reduce the mutation rate again. And it's kind of weird if you think about it, right? Like somehow, obviously, these mutation rate dynamics, it's driven somehow by some balance between beneficial and deleterious mutations. I mean, that must be the case, right? These mutators, they fix beneficial mutations more quickly, and that's why they spread. But on the other hand, they also fix deleterious mutations more, and that somehow limits them, and that's why anti-mutators that reduce the mutation rate can later spread. And somehow these things are kind of in the right balance, and like it seems a little odd that that should be the case, but it does seem to be the case. And it's such that, you know, these guys that have mutator phenotypes, it's not like they jump out to much higher fitness, right? They're just kind of all in the middle here. You can't really tell the mutator from the non-mutator populations based on the fitness data. So, I mean, why are things sort of fine-tuned in that way, or is it really? I don't know. But I think it's a super interesting question. Yeah.
Is that like a general picture? Like if you take any species in any population, you expect to have a mutator?
It is definite, like pretty much everything, yes. I mean, if you just, the thermodynamics of base pairing implies mutation rates of on the order, whatever, 10 to the minus 3 or whatever, depending on the base pairing, right? So, and you probably, you know, if you take any elementary biology course, you learn this, and then there's all kinds of proofreading mechanisms that are required to tune the mutation rate down to, you know, in the yeast, the 10 to the minus 9 to 10 to the minus 10 range that we observe in wild type yeast or E. coli. And so there's a lot of machinery involved in doing that, right? Like you've probably read about kinetic proofreading and stuff like this, right? And so, you know, it just follows that, you know, there's lots of proteins involved in these proofreading processes, and so you can knock them out. And so there's, in pretty much anything, there's a fairly large target size to knocking this stuff out and elevating the mutation rate. And I mean, that's true in humans too, right? Like a lot of, you know, genetic predispositions to cancer are mismatch repair mutations.
I guess I'm just wondering if like if there was some packaging reason or something else why you expect some, like apart from the fact that it can't happen, like
That's a question that, yeah, I mean, you could argue about that. But certainly just like mechanistically, right, given the thermodynamics of base pairing, like you need a lot of machinery to get it lower, and therefore it's impossible to not have this thing. Okay. All right. I'm going to kind of skip around here because I got 20 minutes. I just want to give some sort of highlights. We see clonal interference here too. All right, and you know, we can think about it in this same sort of model. Okay, but another sort of thing that you see in these things is, you know, I talked about this separation between ecology and evolution and how I was going to just sort of ignore ecology. But you look at these experiments and you realize that you better not really do that. Here is an example of one of those populations. If you look at it here, for about 20 years, there's these two coexisting lineages. There's some lineage that has all of these mutations and some lineage that has all of these. And they coexist. You know, it's sort of maybe a little hard to see, but if you're paying attention, you can sort of see it here through this very long time period, okay? And, you know, clearly there are mutations fixing within each type. So there are new mutations arising in this lineage, new mutations arising in this lineage. Occasionally those mutations lead to kind of shifts in the relative frequency of the two types, okay? So like here they're roughly 50-50, here they're like 70-30, here they're like 90-10, whatever, okay? But they coexist for a really long time, okay? And it turns out in some of these cases this coexistence is maintained, well, in all these cases the coexistence is maintained by some kind of negative frequency dependent selection, meaning that a mutation arises which creates a lineage which is more fit than its ancestor if the lineage is rare and less fit if the lineage is common, right? So that leads to sort of a stable intermediate frequency. Okay. And obviously it's sort of totally out of bounds for all these models that I've talked about, right? It implies some fitness that isn't just a number. It depends on the frequency of the genotype as well. So like S is a function of X, as I've been talking about it, okay? And you see that happen all over the place. Here you see it as well. Like I'm just highlighting these guys. This is that original T mutation, and here we have this coexisting two lineages that coexist. I'm marking them by these two mutations that happen later that actually reduce the mutation rate. So this is another sort of thing. All right. And this happens like all over the place. Here are the 12 lines, and we see 8 cases out of 12 in which mutations spontaneously arise that cause this. And it's sort of interesting, like we see this all over the place in E. coli. We don't see it in yeast. I don't know why. Yeah.
Do they interact other than competition for resources?
So we don't really know. So in at least a couple of these cases, it appears to be, this interaction appears to be basically due to acetate cross-feeding. And basically what it is, these strains, like they go into the media, there's a bunch of glucose, or not a bunch, but there's glucose. And so you could imagine one strategy, just eat the glucose as fast as you can. One of the byproducts of this sort of rapid eating of the glucose is acetate. And so, the sort of wild type strain, what it does is sort of will eat the glucose as fast as possible, and the acetate will just leak out. And then they'll later switch to eat the acetate. But you can imagine that creates a sort of niche for some organism that's going to specialize in eating acetate from the beginning. And it won't grow as fast, okay? But it's sort of like the strain that's growing fast, eating all the glucose, inevitably it leaks out the acetate. This other strain is there, like I'm going to munch up the acetate before you can switch. And so that's the sort of basis for this, for this in at least a couple of the cases. But in other cases, we don't know. Yeah.
So what you described right now was essentially you're going from one nutrient to two nutrients, kind of like a couplet. So, but then can you talk about, is there a possibility of having more number of coexisting species than nutrients?
We see one example where it appears there's 3 things that coexist. I mean, I don't know. The sort of remarkable thing about this to me is, right, that I don't know if this is the best model system to study this in. I think there's a good argument it's not. And so I don't want to like go too down in this rabbit hole of studying this phenomena in this system, because they're probably better systems. But the thing that's sort of interesting to me is like, right at the very beginning, I said like, we're talking about, evolutionary dynamics and population genetics, and we're just going to assume the population and, take ecology aside. And, this is the experiment that actually, Rich designed this specifically to study that, with that in mind, and like he tried as hard as he could to think of like conditions in which this kind of stuff wouldn't happen. And yet you see, even in this sort of simplest context that he could dream up, admittedly 30 years ago now, even in that case you can't neglect, ecology and evolution are happening on similar timescales and the interactions are clearly important. On the other hand, in our yeast populations, albeit over shorter timescales, we typically don't see that. So, but it just suggests that, a major sort of, let me put it this way. A lot of times people sort of think, okay, we're doing these experiments to sort of test theory, okay? And sometimes that's the case, but really I don't think that's the utility of these kind of evolution experiments. The sort of main sort of goal for me is we are at a stage where we can sort of observe, we can control parameters in the lab, and we can just observe evolutionary dynamics in really simple circumstances, and we can sort of start to get ideas of what kinds of simplifications we might be able to get away with and still describe something and not. What are kind of inescapable complications and what are not. And that sort of just helps us think, right? We have all these models, we could make them arbitrarily complicated. What simplifications are useful and what aren't? And one of the things I think that we're learning from these experiments lately is that interactions between evolution and ecology occurring on comparable timescales are maybe a sort of inescapable complexity, and we need to think about what are the simplest null models of those processes. And we can try to think of that guided by these experimental results, like what those kind of models would be and what kinds of phenomena we want to describe. And I don't necessarily want to get into it in great detail in this system, but then think about what are the kinds of models, what are the systems to sort of look at those dynamics. And folks like Alvaro Sanchez, for example, and other people are trying to develop model systems for looking at these kind of interactions. Yeah.
Out of the hundreds of mutations that we fixed
Yeah.
Across this 60 or 60
Yeah.
how many, what is the ratio of loss of function to gain of function, or loss of function to total?
So, it's sometimes hard to know for sure whether things are loss or gain of function. Certainly the majority of mutations seem to be loss of function, but it seems likely that some non-negligible fraction involves some kind of gain of function, because you do see mutations repeatedly in essential genes. And so that they really can't be loss of function. So, for example, in our yeast experiments, we see lots of mutations in ribosome-associated proteins. And many of those are essential. And so we believe they're somehow gain of function. But the ratio is sort of hard to know for sure. All right, I have 10 minutes left. I just want to talk about other kinds of questions we can ask. So, another kind of question that we can ask in these experiments is, what does the fitness landscape look like? And this sort of gets again to the sort of point I was raising earlier about, using the experiments to guide, guide simplifications we make in theory, right? So, in all of this stuff that I've talked about from a theory point of view, we've had this sort of row of S, right? And that sort of defines a very simple sort of additive fitness landscape, where each new mutation, its effect doesn't depend on what other mutations you have. Okay, and we know obviously that that's not true in general, but maybe it's a sort of good approximation for capturing essential features of evolutionary dynamics, or maybe it's not. And so a natural thing to do in these experiments is to sort of look at what do the empirical fitness landscapes look like and try to understand them. Okay, and ask, are there any sort of general rules? Okay, and we've spent a lot of time trying to do that. I don't have time to sort of go into all of the details, but one thing we can do, let me just sort of skip forward a bit. Well, probably can't do that. I'm just going to have to tell you the answer. But, one sort of signature that we see in these experiments is there are mutations that happen sort of repeatedly. So these are mutations in this gene ARG-R, which across the 6 populations that don't fix mutator phenotypes, you get 7 mutations. They appear to be some beneficial mutation. Okay? They happen repeatedly, but they're sort of clustered in a subset of the populations. Okay? Which is sort of suggestive of, maybe these are mutations which were not beneficial in the ancestral background, but sort of become beneficial because something earlier happens, right? And there's some famous sort of examples of this. So like, how many of you have heard of citrate metabolism in the Lenski experiments? A bunch of you. So the one sort of very well characterized example of this, there's one of these 12 populations that the E. coli evolved the ability to metabolize citrate under oxic conditions, which is like a major new innovation for E. coli. You know, one of the definitions of E. coli as a species is that they can't do that. And they learn how to do it, and it turns out it involved, you know, some earlier mutation, which then opened up this possibility for some later mutation. And so, you know, that kind of thing suggests the landscape is sort of fundamentally more complicated. And so we can see some signatures of that. One of the kinds of things we want to do in these experiments is characterize that. I won't go into the details because I want to sort of lastly talk about one other kind of direction that we're going in, right? So with the sequencing, we are only sort of seeing mutations that reach a frequency of, you know, 10% or more, which at some level in these populations of 10 to the, you know, 6, 10 to the 7, you know, some of these experiments 10 to the 9 individuals is really only the tip of the iceberg. So lately in my lab we've been really working hard to improve this frequency resolution. And to do so we've been sort of going back to this older approach. In the old days, before you could do sequencing like we can now, what people would do was something like this. You know, they'd like take some yeast and they'd label, you know, with 3 different fluorescent markers, and they'd make a population, they'd mix the 3 different fluorescently labeled yeast, and they'd track the frequency of the fluorescent markers over time. And this kind of, you know, basically looking at the frequency of the fluorescent markers, you could try to infer something about what was going on. But this kind of stunk, you know, because you might have a population, let's say this is a population of 10 to the 8 individuals, so, you know, there's 30 million individuals with each marker, lots of stuff is happening, right? And you're sort of only getting a very crude readout. But what you can do nowadays, okay, is actually have, instead of fluorescent markers, we can label yeast with DNA barcodes. And we can have millions of DNA barcodes, so we could label essentially every yeast cell with a unique barcode. Or in practice, we typically do a couple hundred, which is sufficient for our purpose. And then every mutation that arises, you know, you never get just by chance, because there's only a few individuals with each unique barcode. Every time you get a mutation, it happens in a barcode that has no other mutation. And what it does to the frequency of that barcode over time is essentially a readout of its effect. Okay? And so you can do this. So Sasha Levy and Gavin Sherlock's group and Dmitry Petrov and Daniel Fisher and Jamie Blundell, others were involved in this. They did this in these yeast populations. They started with, this I think was a population of about 10 to the 9. They put 10 to the 6 or so barcodes. So the average barcode labeled a lineage of 1,000 cells. And then they track the frequency of the barcode over time. And they see, you know, thousands of barcodes get beneficial mutations that lead them to increase in frequency. Others go extinct. Okay? And, you know, you can track dynamics this way. The problem with this is that what happens is, showing this as a kind of Muller plot, this is not the same population, this is one of our populations, but you get, you know, lots of barcodes, okay? This is sort of challenging to show in these diagrams because, you know, there's like 100,000 or more barcodes here. I don't have enough pixels, so I'm just collectively showing the frequency of all the barcodes that don't individually reach sufficient frequency to get a pixel as white. But a bunch of them, basically to get a pixel, you got to pick up a mutation and grow, okay? Because there's about 1,000 pixels here, so it's like you got to get a frequency of 10 to the minus 3 before I can show you. But we know the dynamics lower down. It's just hard to see. Anyway, you get all these competing lineages. This one eventually is going to win out, and you can sort of see you can't see anything anymore. So what you would like to do is like subdivide these things. And so that's kind of what we figured out how to do. In our case, every 100 generations, you know, we barcode, we evolve, some of the barcodes increase or decrease in frequency due to mutations they pick up, and then we add a second barcode to like subdivide the lineages. And we do that again and again and again. And so, okay, there's a whole complicated story about how you do this, all right? I mean, these things, like, I mean, you work on this stuff, like you actually have to learn a lot of the biology, or hire people who do. Anyway, And so like then you can subdivide. Okay, so here like we subdivided this lineage, okay, while it was still really small. Okay, and you can see like here are all the sibling lineages after it got subdivided. Sort of tells you something about what was happening within that lineage. And you know, we subdivided again. Now you can sort of start to see these things are mostly equivalent, but okay, some of these sublineages get additional beneficial mutations, right? And you can see that competition playing out, right? And we get, they keep doing all this stuff, all right? We did this a bunch. I don't really have time to tell you about it, all the details, but then what we can do, you know, from these lineage dynamics, we can infer which mutations happen, what their effects are, and you know, you see some dynamics, and from this, like, we can actually like plot the traveling wave. So here's one population. You know, new mutations arise, they're at higher fitness, they grow, new mutations arise like this, okay? Let me just show a video of this kind of. This is the distribution of fitness within the population as a function of time. The black bar is the mean fitness. You can really sort of see all these dynamics happening. And so, all right, I can't show you all of them. But you can see all these kind of jumping of new mutations, all right? And the last thing I want to talk about is, you know, we can start to look at other kinds of more complications. Like, here's an example of a couple populations where we had sort of fluctuating environmental conditions, okay? Same kind of deal. You see a whole bunch of mutations arising. Some of them, you know, they're beneficial in one condition, deleterious in the other, and so they go sort of up and down. And, you know, we can sort of visualize, here's an example of one of these populations. This is the fitness of a lineage in one of the environments. This is the fitness in the other environment. These lines represent mutations, and the dots represent the current frequency at this time point of those mutants. The black line is the mean fitness of the population. These arrows show, like, at this particular moment, which direction is the selection acting in. And so, you know, we can start, obviously it's sort of an interesting question. If you have some pair of environments, or a larger number, and you're fluctuating in some way, and you have mutations, you know, that have some distribution of fitness effects across the two environments, how does the dynamics depend on, you know, what that joint distribution of fitness effects looks like, what time scales do you fluctuate on, what the randomness in the process is, and so on. And so, you know, this is the sort of thing we're just starting to observe. Here's the other replicate population. And, you know, this is, I think, a question about which there's very little sort of theoretical understanding of, for example, you know, how do we expect these dynamics to depend on the time scales at which we fluctuate? And, you know, how repeatable is the dynamics, right? We can do these replicate populations. They look different. How similar or how different should we expect them to look? What does that depend on? All right. So there's a lot of open things to work on. And so, you know, I would say, like, in the future, we can continue to sort of improve on these methods of observing dynamics within individual populations. We also can sort of devise systematic ways of characterizing these fitness landscapes. You know, there are a variety of kind of approaches ranging from things which, like, you know, take some prior knowledge of metabolic networks to methods that sort of think about structure of fitness landscapes, to various geometric models that people tend to like. And, you know, we can sort of look at that by sort of surveying natural variation. There's sort of combinatorial ways that we've been developing to construct landscapes and actually measure them. Another sort of thing that I sort of think is important in this context is to, you know, think about, you know, you can have very complicated fitness landscapes, but landscapes where, even though there's a lot of epistasis, the distribution of effects of new mutations changes in predictable ways. We also sort of want to think about, like, this question of predictability and repeatability of evolution. Here's an experiment which I really like, where this guy Olivier Tenelion in Paris evolved 100 and some E. coli lines for 2,000 generations to high temperature, and he just sequenced them all. It's just like absolutely identical conditions starting from an identical strain, and asked, you know, what are the similarities and the differences in how they evolve. So here, like each of these horizontal lines is an independently evolved population. Each of these marks is a different kind of mutation that happened to occur in that line. And you know, you can see there, you know, the replicate lines, they're kind of similar but also kind of different. And you know, we need some kind of theoretical frameworks for understanding what does this mean, right? Like how predictable, you know, is this like, let me put it this way. What is it that determines how similar or how different these lines are, and therefore what can we learn from these kind of observations? All right, and I'm just going to stop there because I'm out of time, and I should really make sure to acknowledge people that have done, you know, worked with me both on, you know, a lot of this theoretical modeling as well as on the experiments that I just briefly showed you, particularly folks right now who have been the folks that I've worked primarily on with the theoretical work, Ivana, Katya, Elizabeth, Ben, Sergey, Lauren, currently Matt, Jisun a little bit just very recently, and then experimental work. Essentially everyone else has been doing a lot of this experimental work, particularly, you know, Alex has done all of this all these sort of barcoding methods, and earlier work on the sequencing was done, you know, by Dan, by Ben, by Mike, and really by everyone else. So I'll stop there. Thanks, everyone. Yeah.
I guess people ask this a lot, but like you have the constant increase in mutations and the fitness sort of.
Yeah.
So like how would you sort of model that?
So I mean, one thing that appears to be a sort of general phenomenon is there seems to be this sort of general pattern of diminishing returns epistasis, where, you know, there are lots of specific exceptions to this, but on the whole, it seems like as these populations adapt, there's some large pool of mutations, hundreds of genes in which mutations can occur that would be beneficial, but they get sort of systematically less beneficial as the populations adapt. And so, you know, somehow like what you're doing is you're sort of maintaining, there's still lots of beneficial mutations, but they're just less and less beneficial. And so you do get some slowdown in the substitution rate, but it's sort of very weak because that depends only weakly on S, you know. So I mean here, it depends only very weakly on the selective effects. But obviously the rate of fitness increase depends more strongly on the selective effects, and so that may be part of what's going on. Now, why you have this pattern of diminishing returns is not obvious. It's intuitive from some perspectives and then totally weird from others, and I don't really have a good explanation. I don't think anybody has a good explanation yet. It's also true that like there's obviously, as you adapt, there's less beneficial and more deleterious mutations. But that maybe is less of an effect. All right. Thanks, everyone.